Okay, ladies and gentlemen, good morning. Today we are going to look at advanced enzymology, and this small video tutorial was prepared by Mukhtar Aliyu, which is me, and the aspect of this advanced enzymology is going to be look at protein purification, or you can say enzymes purification. Okay, ladies and gentlemen, why do we need to get proteins in its pure form or enzymes in its pure form? Is because know the amino acid sequence. If you are using impure protein, you will end up of getting false positive amino acid sequence because of the presence of other junk protein. And you also need to know, or you also need to get your proteins in a pure form in order to obtain the crystal structure of the protein. Why do we need the crystal structure of the proteins? Is because it is the crystal form of the proteins that are used mostly in rational drug design discovery or in vaccine development. Like for example, unless if you know the crystal of the protein, that will give you the basics idea of the active site of the enzymes, which allow you to select an inhibitor that will be perfectly fit into the active site of the pure enzymes, or that will fit into the functional part of the proteins which render it inactive. And another reason why we need to know we want to get the proteins in a pure format is to know the biochemical functions of the proteins. And it is actually with the few proteins that will allow you to know the evolution of the proteins and the diverse organisms. When you said evolution, it means it is this few protein that will allow you to know how relatedness you are with other living organisms. Then the next thing is the sources of the enzymes or the sources of proteins. Yes, of course, enzymes or proteins are present in huge amounts in animals, but obtaining protein from animals is difficult because sometimes you need to slaughter the animal before you get the tissue or organs in which you are intended to get your enzymes of interest. Then another source of proteins or enzymes is obtained also from plants, but also obtaining enzymes from plant is tedious because it is even more difficult to obtain enzymes from plants than in animals, because plants are highly rich in phenolic compounds which always bind to the enzymes, render them inactive, or even denatured the enzymes completely from the initial steps of enzymes purification. But due to the emergence, or due to the coming, or due to the presence of now area of advanced enzymology, a newly compound was obtained which are mostly used to bind with the phenolic compound during enzyme purification from the plant, which is now make it easy to obtain enzymes from plant. But in the olden enzymology, to obtain enzymes from plant is almost totally impossible. Then another source of enzymes is also You can also get enzymes from microorganisms, but obtaining enzymes from microorganisms is quite simple because they are unicellular organisms and they can't simply or easily excrete enzymes into the culture media by introducing a substrate that will allow it to sense a signals into the system of the microorganisms, making it to be started or making it to start producing the enzymes in a large amount. Like for example, most of the microorganisms are highly rich in extracellular enzymes like chitinases. So sometimes if you want to get chitinases from microorganisms, what you need to do is just to introduce a substrate into the culture media. Like for example, to obtain chitinase from the microorganisms, what you need to do is to add chitin into the culture media as a carbon source, thereby allowing the microorganisms to be excreting the chitinase enzymes to digest the chitin for their own consumption. So as we can see, these are the examples of microorganisms producing enzymes. We have bacteria, fungi, yeast, and mold, and the last one is algae. But remember that fungi and algae are all eukaryotic unicellular organisms, but bacteria are prokaryotic unicellular organisms. But you know that prokaryotic unicellular organisms are much more simple to deal with, and so also at the same time they are also have higher generation time. That is why they can be easily generated in large amount or into a uh, high or large biomass in a culture media, thereby producing your enzymes of interest in large quantity, and their system is much more easier to control than eukaryotic system. And so also at the same time, the productions of the proteins is much more faster because they are mostly operate under operand system. So ladies and gentlemen, thank you for listening. We are going to continue in the next video tutorial on the enzymes purification step. Thank you for listening.